my name is Anthony Cruz, and I will be lecturing on Connectomics. So the age-old question is, what makes us tick? As you can see here, we're kind of visualized as a bunch of gears working together. And I like to think that there's some truth to this thing, and I'll elaborate why. There are two explanations of why we act the way we do and why we do the things we do. So the first one is, the first component is nurturing. So how you were raised, your upbringing, will define who you are. They were saying. So if you grew up in an aggressive household, more than likely you're going to exhibit some aggressive traits. The other component is nurture, nature. So your way your DNA is described, what types of genes you have. So if you have an aggressive gene, then you're more likely to exhibit those traits. So that fuels the nature versus nurture. They're constantly back and forth. But I like to think of a sort of compromise between the two. Right here, you'll see this little girl from the pocket corner, where two families were arguing about whether to have soft or hard knocks. And I like to extend this analogy to neuroscience. So it's not just one component. We can have both. So let's remind ourselves of what DNA is. DNA encodes for everything that you are, from your toenails, your eyelids, to your pinky. And it goes for brain development. But what exactly is encoded there? It's not like you have just a bunch of brain, it's just brain. It's made of a central component, and that component is called a neuron. Its whole structure right here. And there are two major things that you need to know about it. One, the purple body to the left, is called a soma, and the axon, the thing covered in yellow, the longest wire of the neuron, is, it's called the axon. But we aren't made up of one neuron. We're not even made up of two neurons. We're not even made up of 9 neurons. There's a central dogma in biology, and it's not the one in translation. It's more so that cells will make tissues, and tissues will make organs. And we see this right here. So these are neurons, and then it manifests itself into tissue if enough are present. And then in bulk, that tissue will give us a brain. And your brain consists of about 90 billion neurons and almost a quadrillion synapses. And by the way, synapses are just a fancy scientific word for the gap between neurons. And by the way, the quadrillion is 10 to the 14 zeros. So if we really appreciate this method of thinking, we come to realize that the brain is just essentially just a bunch of neurons and how they're connected. We didn't see any other structure in there. So what defines us is the neurons that are in us and how they're connected. And the idea is that function, memory, illness, or anything else we'd like to figure out about what makes us who we are lies within these neurons and their structure. So what are these structures called? They're called connectomics. And how do we see them? We see them in two levels, a micro level and a macro level. So the micro level is depicted right here. It's every single neuron that your brain possesses. And the macro level is just measuring the white matter in your brain, the axons. So the micro level, again, this is a neuron, and that's a cube of neurons. And those neurons are stuck into that cube. And that cube is just a couple microns small in volume. And that's insignificant to our brain proportion. So that cube, in comparison to our next picture, that's even more insignificant. And if we really wanted to, we could count all those individual neurons. And even this picture of just endless branches, endless neurons that you can see here, doesn't even stack up to what we see here. So the cons are it's very, very daunting. And right now, we can only see these individual neurons if we color in those neurons manually, scan them manually. And there are billions more uncounted. Even if we counted those by hand, we'd still have billions more uncounted. And the pros are, the reason why we embark on this endeavor is that we think it will lead to a precise diagnosis or a precise answer as to where abnormalities originate. The second type is the macro-connector. And this section of the brain is just the brain cutting out, coronal section. In the front view, you can see white matter and gray matter. Gray matter is pink stuff, anything. And the white matter is white. So the white matter contains primarily axons. Remember those nerve fibers, the things covered in yellow. And the gray matter is primarily soma, the bodies of the sun. So right there, the body of the sun. And they're made using diffusion spectrum imaging, diffusion SI. And what it does is it collects the water in the axons and it makes it go in a certain direction. And because they're all going in a certain direction, we can generalize the structure using some sophisticated algorithms. So here's the antibody. And the pros are, these can be made within minutes. We have the technology right now to make them. And they could tell us enough about why we are who we are. The cons are, we think that these might not be ultra specific. Although they've not been used in practice yet, we fear that we may run into some dead ends, that these aren't going to be able to tell us enough. So they may not tell us who we really are. So how do we use this? So the idea is that we'll compare structures. So say these are two different connectomes or two different people. One has a mental abnormality, the other one does not. So what we do is we compare these mathematically, if you will, and we find a probability difference between these structures. And that probability difference will tell us enough about the person so that we can say, this is the root of the problem. I'll just explain that. And thereafter, when we see where the origin of the illness is, we can apply treatment. So maybe injecting stem cells or producing growth in that area. And a comparison would be genuine. So this is a normal brain structure. As you can see, everything's intact. And abnormal brain structure is missing some things. So in this way, we can think of Connectomics, of mental abnormalities. There's structure, and then there's differences. And those differences produce who we are. So our hopes for the future are that microconnectomes will be formed, and they'll be able to tell us more about ourselves. And mental illnesses and things of the old will be explained. And in turn, we'll have a healthier population. So I want to end with a note that in February of 2001, we completed the Human Genome Project. And we thought that this would tell us everything that was to know about ourselves. And it did. It told us a lot, but not the whole story. The truth is that the connectome is the missing link. That nature and nurture do work together. That your genes code for how your connectome initially forms, and then environment takes place. That these two work in combination, these coexist to make you who you are. That they form your structures, and the structures define you. Thank you.